Yeah, so right after class.
Yeah, that'd be great.
Thank you.
All right.
I have a question, Melissa.
Sure.
I don't know if you know. Do you know how to do a natural log on this calculator, or can you?
Yeah.
Because when I do that, it
Oh, okay. So do
gives you an
this
error. I don't know if you have to do it.
and then do that.
10.
So yeah, do the number first and then hit natural log.
Okay.
Yep.
Cool. Thank you.
You're welcome. All right, everyone. Good afternoon. Hello, hello. All right. So today your problem set number 2 is due. I do have a few students that would like to maybe talk to me after on Friday about the problem sets. If you want to stay after, I can draw on the board or, really just remember these are, we want to have these due dates to keep you on track. These problem sets are going to help you with your exam, but these are soft due dates, so if you're still really unsure, I can bring them on Friday. We can talk about it. You can adjust your answer. Remember, your score is really participation. Okay? All right, but remember these are important because they're going to help you study for the exam. All right? Okay, so we are recording. I want to just make sure that everybody knows that we are recording at this point. Today what we're going to do is we're going to start with passive, facilitated, and active transport. So we're really going to slow down a little bit now, okay? So I understand that, boy, we've been really, with all of these problem sets and trying to understand equilibrium potential and resting membrane potential, these are difficult concepts. Now we're actually going to slow down a little bit, okay? So I think that this one is a little bit more conceptually easy. No less, it's very important still, so don't, take it for granted. Still study for it. But I think that you'll enjoy just kind of the slower pace. Okay, any questions so far about your homework assignment or equilibrium potential or anything like that? Okay, before we get started with the lecture though, I do want to answer a question that a student had after class on, what is today? Wednesday? After class on Monday. And I thought this was a great question. The question was, how do I think about driving force and conductance? Okay, how do I think about that and put it in perspective to a steady state? So you can take a picture of this with your iPhone after I draw this on the document camera here, but if you want to take a look, if everybody wants to take a look around the room, I'll turn on all the TVs so that you can see what I'm talking about. All right, so we have been calculating equilibrium potentials. I think now we can discuss this because this was still a little bit, fuzzy for everybody. So let's just say the equilibrium potential for sodium is about plus 61 millivolts, okay? Equilibrium potential, you already calculated it out. Equilibrium potential for potassium is minus 80 millivolts, okay? And when we actually measure the resting membrane potential, let's just say this is about minus 70 millivolts. And that's a solid line because it's an actual measured value, all right? The reason why it's so close to the equilibrium potential for potassium is because if we use the chord conductance equation, right? Everybody kind of knows this now. To predict the membrane potential, right? Then we would see that basically out of all of the total conductance, about 90% is usually due to potassium. Only 10% is due to sodium. So if we calculated this out, it would be about minus 70 millivolts. Is everybody with me on that? Okay, if it was 50-50, the resting membrane potential would be up here, right in between the two. If it was 90% sodium and only 10% potassium, it'd be way up here, closer to the equilibrium potential for sodium, right? I hope that makes sense to everybody so far. All right, so how do we think about driving force? All right, the driving force, you'll remember, is equal to Vm minus Es. That's for any solute. So for sodium, it has a huge driving force, right? Vm minus ENA. That's a huge driving force. And for potassium, ooh, it's kind of small. Vm minus EK. Tiny driving force. Okay? Now, at steady state, remember this resting membrane potential is staying steady. Do you remember my analogy of the infinity bowl, that bathroom? You walk into the newer houses, the influx of water has to equal the efflux of water to keep that water line steady, right? So let's put some numbers to it, or equation to it. What I'm saying here is the influx for sodium, the current for sodium, has to equal the current for potassium in the opposite direction. And this is steady state. Okay, everybody following me so far? All right, so with that, you all know that the driving force, this is just Ohm's law. The driving force is equal to current times resistance, right? So you can agree that I could write this, right? I could just divide this by R, and the current is equal to driving force over resistance. Everybody with me on that? Okay, so I'm going to do one more switcheroo, and current is equal to driving force times conductance, because resistance is the inverse of conductance. All right, so now I can actually write this way. Instead of the current for sodium is equal to the current for potassium in the opposite direction, couldn't I write it this way? The driving force for sodium times the conductance for sodium, that's just current, is equal to, but in the opposite direction, the driving force for potassium times the current for potassium. Right? I said the same thing. Current for sodium is equal to driving force times conductance is equal to driving force times conductance for potassium. All right, so the reason why they're equal to each other is because the driving force for sodium is large, but the conductance is small. It's the opposite for potassium. The driving force for potassium is small, but the conductance is large. That's why they're equal to each other. That's why they're at steady state. Okay, so going back to here, conductance is large for potassium. That's why it's closer to the equilibrium potential for potassium, but the driving force is small. For sodium, the conductance is small, but the driving force is large. All right, for those that are still a little confused, let me give you this visual. All right, this is maybe how you can think about it. I like to think about it in hydraulic terms. So let's just pretend we have a plasma membrane, okay, and 9 out of 10 faucets. We're going to use hydraulic terms. 1, 2, 3, 4, 5, 6, 7, 8, 9. Oops, and I'm going to do 9. 9 out of 10 are, if this is inside the cell, and this is outside the cell, 9 out of 10 faucets are open, and they're conducting potassium out of the cell. Right? 1 faucet is conducting sodium into the cell. Now the faucets are the pathways, so you can think about that like conductances, right? 9 out of 10 conductances are due to potassium. only 1 out of 10 is due to sodium. But the driving force, remember the driving force for water is that pressure gradient. Once this sodium channel opens, it is spewing sodium out. Just imagine like when you turn the faucet and you don't realize that the pressure gradient is so large that it just gets all over you, and I mean it spews out. But you turn on all 9 of these potassium channels and the driving force is small. So it's just like trickling out, but the currents are the same. The influx equals the efflux. So that's how you should think about the driving force and the conductance. Does that help? Does that help everyone kind of solidify that? Okay, so that's why you are calculating driving force in addition to knowing the conductance. Pretty good? All right, any questions? I just like to give everyone that visual because it helps me. I mean, it helped me when I was a student, really understand it. Okay, so aside from that, we are actually now going to start our lecture on passive transport. We're going to do a top hat question, but it's going to be a little bit later in the lecture here. I'm going to try to get through passive transport, passive diffusion, and facilitated diffusion. Okay, so go back to our document camera here. There's only a few slides for passive diffusion, but there's a lot of information in these few slides. I do have to apologize. I haven't posted last Friday's or Monday's lectures yet. They will be posted today. They're already on YouTube. It's just a matter of just putting those links on Canvas. So I'll do that today along with this one as well. All right, so skipped ahead here. We're talking about passive diffusion. Sometimes passive diffusion is called simple diffusion as well. Passive diffusion can also be called simple diffusion. All right, here's your set of learning objectives. Again, when you get home, you can take a look at this. We're going to start off with the first learning objective, identifying sources of energy, driving the process in the molecular pathway for diffusion, facilitated diffusion, and then we'll get to primary active and secondary active transport. So just overall, I want you to take a look at this particular diagram. There's 3 modes of membrane transport that we're going to be going over. Passive diffusion right here, passive diffusion, it doesn't need a protein, right? These are usually hydrophobic molecules that can easily enter into the plasma membrane and cross the plasma membrane easily, all right? That's called passive or simple diffusion. No protein is required. Facilitated diffusion, like when you have transport of ions, electrolytes, or larger molecules, you can actually see that a protein is required. Now, for all 3 of these, passive diffusion, in this case, facilitated diffusion including ion channels and permeases, note that always, I shouldn't say usually, always, these proteins or passive diffusion, the net diffusion is always going to go from high concentration to low concentration. Or in other words, it's always going to go with its concentration gradient or down its concentration gradient. And no energy is required. However, when we get to active transport, you'll see that you do have to inject energy into the system. See, this is the concentration gradient. High on the outside, low on the inside. But for active transport, it harnesses energy, say from the hydrolysis of ATP, to move molecules against their concentration gradient from low to high. Okay, so the take-home message for this slide is that transport mechanisms are distinguished by direction of transport, nature of the carriers, and the role of energy. Okay, so let's concentrate first on passive diffusion And what I would recommend, just, if you have a spare piece of paper there, I would just write passive diffusion, facilitated diffusion. Just start this little note, and then active. Okay. So, and then you can put some of the characteristics there. We already discussed this. No protein is required. You see where I'm going with all that? If you can just build this chart, it might be a little easier when you go back to study. Okay? So just a tip. Let's go back to our slide though. All right, so taking a look at this, you'll notice that when we're talking about passive or simple diffusion, usually what we're talking about are these small hydrophobic molecules like gases, oxygen, carbon dioxide, nitrogen, benzene. That's not a gas, but so basically these small hydrophobic molecules, they can easily cross the plasma membrane without any protein. No protein is required. Also, small uncharged polar molecules like water, glycerol, ethanol easily move across the membrane. Now, we did talk about water channels. I don't know if you remember that. Water does move across the membrane, but if you really want to facilitate water moving across the membrane, water channels actually really increase the amount of water going in and out of the cell. All right? So now you can notice that large uncharged polar molecules, that includes glucose or ions, they can't go across the membrane. They really do require protein, and so we're going to put them in facilitated diffusion. So this is just giving you some characteristic of some of the molecules that do move across the membrane by passive or simple diffusion. Okay? So now let's add on. Let's ramp it up a little bit, all right? Like my Pilates class. Adding on. All right, so let's talk about permeability. In your textbook, you probably will notice permeability. You can imagine this was part of Fick's law, but let's elaborate on this a little bit. Let me just remind you what permeability is. I told you that permeability and conductance are very similar, but they are not synonymous. They are not equal to each other. So, I'll tell you again, conductance is the ease at which a molecule moves across the entire membrane from either inside to out or outside to in. Permeability is the ease at which a molecule just enters into the inner part of the plasma membrane or a pore region. You can see that there's just a subtle difference. How easily do these molecules enter into the plasma membrane interior? Not all the way across, just into the plasma membrane. Now, I've added here what's called the partition coefficient. This beta is called the partition coefficient. D is diffusion coefficient. You all know that now. D describes the solute and solvent characteristics. So D is describing the solute and the solvent of a solution. Delta X is the membrane thickness. You remember that from Fick's law as well. So permeability is actually equal to the partition coefficient times the diffusion coefficient over the membrane thickness, in which basically the partition coefficient describes a molecule's ability to partition into either oil or water. So this is how you think about it. The partition coefficient for any molecule, X, if it is greater than 1, you can see it prefers to be in oil rather than water. So if you take glucose, for example, and you measure how, you have this big oil globule, and it's in an aqueous solution, or it's in pure water, and you can isolate both, and you can see where glucose prefers to be. If it's greater than 1, then it prefers to be in oil. If it's less than 1, then it would prefer to be in water. And the beta coefficient doesn't have any units, It's either just greater than 1 or less than 1. Okay? Any questions so far? All right, so let's go to this one. Again, some description at first. We've kind of talked about this already, but I'll give it to you formally. How do we think about selectivity for non-electrolytes? This is determined by the molecular properties responsible for the partition coefficient. Usually it's the molecular size of the solute. The smaller the size, the more it's going to be easier to partition into the membrane. The lipid solubility, hydrophobic molecules tend to easily move into the membrane as opposed to lipophobic or hydrophilic molecules. And then the degree of ionization. If it has a charge associated with it, it's not going to easily move into the membrane interior. Okay? So, thinking about permeability, right? I'm going to step you through this diagram at the bottom. All right? So, J net, this is how your book describes it, is the same exact thing as F net in Fick's law. So that's the flux. Instead of J, sometimes you'll see it in other journal articles as F net. And you already know how to think about this. This is the net flux in moles per second, just like we learned in Fick's law. All right, in this case, the concentration outside the cell is greater than the concentration inside the cell. And this interior portion is the inside of the plasma membrane. Everybody with me on that? All right, we're just assuming this is a linear relationship. This class doesn't require calculus, so we're not going to do anything fancy, right? But what does this mean? The beta, which is the partition coefficient, equals 1. How do you interpret that? How do you interpret that if the beta, or partition coefficient, is equal to 1? Does it prefer to be in oil, or does it prefer to be in water? If the beta, or the partition coefficient, is equal to 1. What's that? That's perfectly right. The answer was it doesn't have a preference. It doesn't prefer to either be in oil nor water. It is equal. So the beta, or partition coefficient, is 1. No preference. All right, with that, you can actually see that there's a certain net flux, depending on this concentration gradient. Everybody with me? Okay, so let's go to the middle panel here. All right, the middle panel, now our partition coefficient is greater than 1. You all remember that that means that this molecule prefers to be in oil. So what you'll notice is, the concentration outside and the concentration inside are still the same, but when you look at the interior of the plasma membrane, you'll notice that the concentration bumps up quite a bit in that inner leaflet, right? Because it prefers to be in the interior of the plasma membrane where that's a lipid environment. So the concentration is greater right in that inner leaflet. The concentration gradient now is steeper. And now, thinking about Fick's law, with the steeper concentration gradient, you're going to get a greater net flux. That's why that arrow is a lot more thick. Makes sense, right? All right, let's do the opposite. Now the beta coefficient is less than 1. This molecule prefers to be in an aqueous environment. It's hydrophilic. It's water-loving. So concentration outside, concentration inside are the same. However, now it prefers to be in the aqueous solution outside of the plasma membrane. And you can see right in that inner leaflet, the concentration really drops. Now the concentration gradient within the plasma membrane is a lot more shallow, and the net flux drops off. Make sense? that helps you think about that partition coefficient and how it fits in to simple or passive diffusion. All right, so let's take it a little bit further. In this case, when we're looking at passive transport, we're only concentrating on the green line here. So let me walk you through this graph. Sometimes graphs can be really difficult to read. What this is saying on the y-axis, this is the flux, net flux into the cell. On the x-axis, I don't think this is labeled that well. This is actually the concentration gradient. So the higher the concentration gradient, the greater the net flux. And you can see there's a linear relationship here with passive diffusion. So in passive diffusion, the flux rate is limited only by the concentration gradient. The greater the concentration gradient, the greater the net flux. All right, so if you are starting that chart that I was talking about, here's a review slide. For passive diffusion, number one, no energy is needed. Number two, it depends on the concentration gradient. The greater the concentration gradient, the greater the net flux. And the direction of flux is always going to proceed from high concentration to low concentration. Steeper gradients results in higher rates, I already said that. And saturation does not occur. We haven't even talked about saturation before, but we'll get to that with facilitated diffusion. That's the ability, basically, when you are actually limiting the net flux into the cell. You have a max flux. But that doesn't happen with passive diffusion. All right, so remember, saturation does not occur. All right, that's it. That's it for passive diffusion. All right, so let's go to facilitated diffusion. Facilitated diffusion, one of my favorites here, because I really, really enjoy ion channels. They're awesome. Okay. So again, here's our roadmap. We just talked about simple diffusion. Now we're at facilitated diffusion. We're going to talk about ion channels, voltage-gated sodium and potassium channels. I'll mention CFTR as well, and permeases or carriers. Right? On Friday, we'll get to active transport. Again, here are our learning objectives. I'll let you take a look at this when you get home. And let's start with facilitated diffusion. Now we're actually talking about ion channels and permeases. Okay? So here's an ion channel. It actually conducts molecules. Mainly, we're going to be talking about ions specifically, but also there's water channels. Okay? And then permeases, the classic example of a permease that we're going to be talking about is the glucose transporters. These are carriers. So once a glucose molecule binds to this carrier on one side, there's a conformational change, and then it releases the glucose molecule on the other side. All right, so for facilitated diffusion, a protein is required. It can't cross the membrane by passive diffusion alone. However, no energy is needed. In this case, it does depend on the concentration gradient, and diffusion is always going to precede the net diffusion from high concentration to low concentration. But in this case, saturation is possible. So take a look at this. What might determine the value for maximal flux, as shown in this slide? Here's the maximal flux. Why is there a max flux? Why isn't it unlimited like passive diffusion? Why do you get to the point where you actually can't exceed this maximum flux into the cell. Yeah? That's exactly right. Okay, so number one, you only have a finite number of channels or transporters. And if all of them have a molecule attached to them or have a molecule inside that pore region, then you could reach your maximum flux. Okay, so the number of channels and transporters are important. Let's just think about a carrier for a minute. What else would also contribute to that max flux? Not just the number of carriers, but what else? Thinking back maybe to biology or... Perfect. Where's that coming from? Okay, table 11, nice job. Okay, so remember each one of those transporters has a turnover rate, right? So if you have a maximum turnover rate, right, they can't go any faster than that. So it's the number of channels that are important or number of transporters and that turnover rate. Okay, all right. So here's a question for you. Facilitated diffusion carries impermeable solutes across the membrane on protein carriers, including ion channels. So let's get your top hat devices out. This is just one question that I have for you for today. Thank you all for coming to class. Here is your question. Just a simple one. It's actually pretty tricky. Are ion channels saturable? Now, we have lots of ion channels. They conduct a lot of ions, way more than carriers. We're going to learn that 10 million ions can move across the membrane through a channel per second, as opposed to a carrier, only 1,000 molecules per second. Do you think we can saturate out an ion channel? I don't expect you to know this, just kind of one of those thought questions. All right, we have about 76 people. I'll go ahead and give you 10 more seconds. We're already up to 90 almost. You can talk to your neighbors. We talked about carriers having a max flux, the number of carriers and the turnover rate, but what about ion channels? All right, I'm going to go ahead and close this one. And there's really no correct response, but I'm going to say yes. All right, here's why this is super tricky. In your textbook, it says no. You can't saturate out ion channels. However, I do know of instances in papers that have been written where if you make the concentration gradient steep enough, specifically for calcium channels, calcium conducts in a certain way in the channel where they line up single file. So like 3 of them are in the channel at one time, and you can clog the system and saturate it out. So your book says no, but I'm saying yes, all right? So there was no correct answer. Thank you for participating. But it also gives you an idea of saturation and how to think about that. So let's talk about ion channels. Okay, here we go. I'm going to start with this one. The reason why I'm not putting it in presentation mode is because I do have a few videos here. Okay, so I specifically really like ion channels. I would actually consider myself more of an ion channel physiologist. That was what my graduate work was in. I studied purinergic receptors. So I just want to give you some information, some facts to hopefully you guys admire and respect ion channels as much as I do. But you can honestly move an enormous amount of ions across a channel. When you're comparing that to a carrier, it's not even close. So proteins, this is what ion channels are. They're proteins that span the membrane. and they allow for a flux of about 10 million ions per second. Again, as I stated before, the pump itself, that's our sodium potassium ATPase, only transports about 1,000 ions per second. That's 10,000 times less. And ion channels are really important. We're going to be talking about them a lot more in excitable cells like neurons and muscle, but they're also important in hormone secretion. Basically, every single cell in your body has ion channels. All right? So there's not ever a situation where there's no ions moving across the membrane. All right. First of all, there are 3 properties that we're going to go over today. We're going to talk about conformational states. That's conductance. See what I did there with the slide? I was just, that first bullet point, we're going to be talking about conductance. All right? Then we're going to be talking about how ion channels open and close. That's called gating. Okay? And then finally, I'm going to be talking about selectivity at the bottom there. All right? So these are 3 properties that I'll be discussing. Please note that that is conductance, number 1, gating of ion channels, number 2, and selectivity. All right, I'll get to selectivity, but I want to just show you this video. This was a student project, not in my class, but I thought this was super cute. Again, it's kind of like your video project. They were trying to illustrate or depict what selective permeability was. Okay, so this is selectivity. And I don't know why it made me laugh out loud, but it did. Literally LOL. Okay.
I'm a semi-permeable membrane. I let some things in, and don't let other things in. For example. No, you cannot go ahead. You, go ahead. No. There. See how I let Laura in, and I did not let him in.
Okay. All right. I like this for a number of reasons. First of all, I like the molecule that goes, ha ha, when it gets to go across. And then I like that the other molecule fights back. So anyway, I thought this was funny. Okay. My own humor here, I guess. All right. So remember, that is one of the properties, selectivity. Most ion channels actually exhibit selectivity. But let's start with conductance first. What does that mean, conductance? All right. So a lot of ion channels have a conducting state and a non-conducting state. Okay, so, and this all depends on conformational changes in these integral proteins. Okay, so we're going to talk about what are some of the stimuli that cause a conducting or non-conducting state. But here's the kicker. Here's what I want you to really get from this slide too. Usually the open state, we know that is a conducting state. That actually allows for conductance of an ion. But there are several different non-conducting states. And when we talk about voltage-gated sodium channels, we're going to talk about closed states and inactivated states. Both of them are non-conducting states. Okay, so just keep that in mind. This is what I mean with conductance. So, part of the ion channel properties, channels assume different conformational states, conducting and non-conducting. All right, let's go straight to gating. All right, this is our second property. There are different ways to open and close various ion channels. We're going to talk about 4 here. All right, voltage-gated channels, they open when the membrane voltage changes. So at rest, you can see the inner leaflet, that's the yellow portion here, that's the inside of the cell, it's got a negative voltage, membrane voltage. But if you impose, right, you depolarize the cell, that's why there's a positive sign inside the cell now, you can force this channel to open. This is called a voltage-gated channel. It has a transmembrane segment that is all positive residues. So when the membrane becomes positive, it forces that residue, that transmembrane spanning domain, up like a corkscrew, and it opens the channel. Okay, that S4 region. All right, that's a voltage-gated channel. The next one is a mechanically gated channel. It's also called stretch-gated or tension-gated. Now take a look at the bottom here. You can see this channel is actually attached to cytoskeletal elements, okay? So if I took a pipette, or you can imagine a pencil or something, and I carefully pressed from the outside into the inside, and I made a little dimple, like I pushed it physically, made a little dimple into the cell, I would stretch these cytoskeletal elements and I would open the channel. That's what's called stretch-gated or tension-gated, also called mechanically gated. All right, so let's think about this. What happens when I drop a cell into a hypotonic solution? What's going to happen? It's going to swell, right? Water's going to go into the cell. It's actually going to stretch these cytoskeletal elements and open channels to try to self-regulate. It's going to start to move solute out, trying to get water to go out at the same time to try to keep the volume in a homeostatic range, right? That's called regulatory volume decrease, okay? All right, so that's another type of ion channel, stretch-gated, tension-gated, or mechanically gated. All right, next one, phosphorylation-gated. You can see with this channel, if you attach a phosphate group, that opens the channel. We're going to be talking about CFTR. This is the Cystic Fibrosis Transmembrane Conductance Regulator. Our next problem set is going to be about cystic fibrosis. And this is actually a chloride channel. When it doesn't get traffic to the membrane, it's absent in the membrane, it causes cystic fibrosis. Again, it's an anion channel. It conducts chloride and bicarbonate. We'll talk more about that a little bit later. But it is phosphorylation gated. CFTR. All right, the last one here is ligand-gated channels. Sometimes they're called ligand-gated receptors. They are ion channels if they're ionotropic. So in this case, we have a ligand, like a neurotransmitter, that binds to the receptor, and that receptor happens to be an ion channel. So it opens up, allowing for ions to move across the membrane. Classic example is the nicotinic acetylcholine receptor, which is an ion channel. It is responsible for initiating muscle contraction. So when we get to muscle, we'll revisit this. Nicotinic acetylcholine receptors are ion channels. For our purposes here, we're just learning about the different gating properties of different ion channels. All right, so yeah, question?
For phosphorylation gated,
Yeah.
could that not technically require some energy, or is it just mean that there's an energy per molecule?
Yeah, that's a really good question. Let me think about that. I don't think that it does require energy, but you're right. It's kind of like kinases phosphorylate stuff. And let me think about that. Let me think more carefully about that. If ATP is required or if it's not required, my inclination is no, but I may be wrong. Yeah, great question. Hadn't even thought about that being requiring energy. Actually, with CFTR, it does use ATP. That's right. So that would require hydrolysis of ATP, and that would require energy, yes. Okay, good question. Yeah, I'll revisit that, give you an answer on Friday. All right, let's revisit the voltage-gated sodium channel. Now, the reason why I'm having you go through these different states, this is still called gating, is because when we get to neurons, this is really going to be important to understand. So, we're specifically talking about the voltage-gated sodium channels. They have 3 different states. They have a closed state, that's what C means. They have an open state, that's what O means. And they have an inactivated state. Now, think about this. The inactivated state and the closed state are non-conducting states. Only the open state is conducting. Now, a few more terms. If you go from the closed to the open state, then that's called activation. That's the transition to a conducting state. If you basically, when the voltage-gated sodium channels open, they almost immediately inactivate. So if you go from the open to the inactivated state, that's called inactivation. And then the channel actually has to recover from inactivation. That's when it goes from the inactivated state to the closed state. So you're probably thinking, why do you have 2 non-conducting states? Seems redundant, right? But they have very different properties. And when we talk about refractory periods, when we get to neurons, this is the key to understanding it. So hear me out here. When the voltage-gated sodium channel is in its inactivated state, it doesn't matter how strong a second stimulus is. You cannot open the channel. There's just no way you can open the channel if it's in its inactivated state. The channel actually has to recover and be in the closed state. Then you can stimulate it and it can move into the open state. Does that make sense? That's what's responsible for the refractory periods in the neurons, okay? So if you go to this animation, it's actually pretty cool. Let's take a look at it. This is going to give you a lot more information about the voltage-gated. Here we go. Let me blow this up a little. Okay. So it's a nice animation. We are actually concentrating on the voltage-gated sodium channel on the left-hand side. Okay. So this M gate right here, this is actually called the activation gate. All right. This H gate is the inactivation gate. Okay, so what's going to happen is when this is actually in the closed state, when the channel is stimulated to open, this M gate opens, sodium rushes in. Then it inactivates, this H gate closes, and then it has to recover from inactivation. H gate opens, and then it's back in its closed state, and now you can stimulate it again. Let me just show you an animation. You stimulate it to open, doom. Now you've actually got inactivates, and then it has to recover from inactivation before it can open again. Now it's in its closed state. One more time. Opens, conducting state, inactivates, and then it has to recover from inactivation back to the closed state. Does that make sense? Yeah, great animation. Love that animation. All right, so that's how to think about the voltage-gated sodium channel. We are out of time. All right, so I will see you on Friday. We'll finish up facilitated diffusion, talk about carriers, and then we'll get into active transport. All right, have a wonderful Thursday, everyone. I'll see you on Friday. Yeah, don't forget to turn your homework